Okay, welcome to Fatty Acid Synthesis Part 1, Take 4. So I have had several challenges videoing this. It turns out due to the fact that my iPad memory was full. So it has been only recording parts of it. So unfortunately, that means that this Part 1 will actually be broken down into an A, B, and C piece now that I have solved my issue. The good news, though, is that every time I've gone through it, I've gotten a little bit smoother at presenting this. So that part will be great. So let's take a look at where we are. We have talked about metabolism. We have talked about fatty acid degradation. And now we want to talk about synthesizing fatty acids for storage. Most fatty acid synthesis occurs in liver cells and fat cells. It is a fairly minor pathway because generally you consume enough fatty acids in your diet to meet the metabolic needs of the body. So some exceptions: if you consume more energy than you need, excess carbon consumed, then the excess is going to be stored as fatty acids. And so this would be the process that your body would undergo to form those. Another example might be something like mammary glands in a nursing mother. There would be lots of fatty acid synthesis with that. So when we talked about fatty acid degradation, we started with our fatty acids. And we came down kind of this route and ended with acetyl-CoA. So as we're talking about synthesis, we're just starting with that acetyl-CoA. We're coming down here through malonyl-CoA, which is like an activated version, and then we're dropping back up this way. So a little bit different. Okay, let's take a look, orient ourselves with this picture. We have been talking about energy metabolism. So we've been talking about taking those carbs through glycolysis, forming pyruvate. From pyruvate, we got the acetyl-CoA. We did the citric acid cycle. And then we shuttled those electrons out to the electron transport chain. So lots and lots of energy, right? Catabolism. We also looked at the acetyl-CoA forming ketone bodies. That was another pathway that we've looked at. But let's think about what would happen. So if our energy needs are met, we don't need to run this citric acid cycle, and we don't need the ketone bodies. That citrate right here would start piling up, okay? And when that happens, it'll make its way out of the mitochondria through the shuttle. This is what kind of a starting point for us, a little bit of a starting point for that fatty acid synthesis. So we're only going to do it when energy needs are met and when we don't need to make those ketone bodies. Now we're going to use this acetyl-CoA. We're going to have to shuttle it out as citrate to form those fatty acids. When we looked at beta oxidation, that was that breakdown of the fatty acids step by step, like 2 carbons at a time. That occurred in the mitochondria. But fatty acid synthesis is really going to occur out in the cytosol. So we got 2 reciprocal pathways separated essentially by this organelle. So fatty acid synthesis takes place in 3 stages. Okay, the first stage is transfer of that acetyl-CoA out of the mitochondria. So we got to get it out into the cytoplasm. We'll look at how that's done. The second stage is the activation of acetyl-CoA to form that malonyl-CoA. Okay, that's the form we need, or one of the forms we need. We actually need both. And then the third stage is essentially repetitive addition and reduction of 2 carbon units. So just like we broke down fatty acids 2 carbons at a time, we're going to build them up 2 carbons at a time. And synthesis is going to occur on what we call an acyl carrier protein. So we'll take a look at that. So first, let's look at stage 1. Let's talk about this transfer of acetyl-CoA out to the cytoplasm. There is not a carrier across that mitochondrial membrane. So let's orient ourselves. So we're in the matrix over here. We're in the cytosol over here. We've got our inner membrane, our outer mitochondrial membrane. Okay, and so we've got our citric acid cycle running in here. And what we need outside is acetyl-CoA, but there is no carrier across those membranes for acetyl-CoA. So instead, it's going to get carried out as citrate, which does have a carrier. Okay, so that oxaloacetate will react with acetyl-CoA just like in the citric acid cycle to form the citrate. But now the citrate can either continue through the citric acid cycle, if we need that energy, or it gets carried out into the cytoplasm. Okay, once it's here, it gets split back apart into that acetyl-CoA and our oxaloacetate. Okay, the enzyme that is used for that is actually written down here, ATP citrate lyase. So let's think about that. Citrate was our substrate, and ATP is the other one. So this tells us that this took energy, right? We needed to use ATP to split these apart. So now, yay, we've got our acetyl-CoA out in the cytoplasm where we need it. But unfortunately, we still need to get that oxaloacetate back into the matrix because we need to be able to continue the citric acid cycle or the formation of citrate to get more acetyl-CoA out here. But just like the acetyl-CoA, there is no specific transporter for oxaloacetate. So instead, we're going to have to get it down to pyruvate so that we can transport this in. All right, to do that, first we're going to have a reduction. Okay, so it's going to be, reduced to get down to the malate. And notice we're using up these electrons, okay, because it's reduced, so it's going to gain electrons. We're going to get those electrons from NADH. And then we're going to oxidize that malate, okay, to pyruvate. And notice that when we do that, if we're going to take electrons from malate, we have to give them to something, and it forms that NADPH. So if you remember when we talked, we said NADH usually used for catabolic functions. NADPH is the one we usually use for the anabolic functions when we're building up molecules. So this is actually really good for us in that we're going to need energy to build these fatty acids, and here's the start. Okay, the net reaction is shown down here. Real quick, let's look at the fact that we need to integrate these various metabolic pathways. So to make the fatty acids, we're going to need acetyl-CoA, some of which will switch over to the malonyl-CoA, and we're going to need NADPH. And we just saw how we got NADPH, but in that process, we only get one NADPH per acetyl-CoA. That is not enough to drive our fatty acid synthesis. So we also are going to need the pentose phosphate pathway. Okay, we're going to have to harness some of the glucose in the body and make sure it gets over here to this pentose phosphate pathway to give us more NADPH. All right, onward to stage 2. Formation of malonyl-CoA is committed step in fatty acid synthesis. All right, so it's rate limiting, it's regulated. Let's take a look at it. Malonyl-CoA is synthesized from acetyl-CoA by acetyl-CoA carboxylase 1. That's the enzyme, and we're going to abbreviate that ACC1, and that'll be an important one that we'll come back to. Okay, and then another piece here, it's biotin requiring enzyme. So let's think about that. Biotin is a carboxylate carrier. Okay, so it carries carboxylate groups. Okay, it's B7. And this biotin is bound to the enzyme. So this reaction, stage 2, occurs in 2 steps. So if you take a look here, we have this enzyme that's bound to the biotin. We said it's a carboxylate group carrier. It's going to grab a carboxylate group from that bicarb, and this is going to take energy. So then, and there's a little typo here, this should still have the enzyme. So we still have the enzyme bound to the biotin, but now over here on the right, it's grabbed that carboxylate group. Okay? Then we're going to see in the second step that it takes that carboxylate group and reacts with acetyl-CoA to add that carboxylate to the acetyl-CoA. So look at the pictures down here. We have our acetyl-CoA. Okay? We have this bicarb, and we're going to add that carboxylate group. So that enzyme added that carboxylate group. So the black part would still be acetyl-CoA, right? But now for malonyl, we have that carboxylate group. All right, and with that, we're ending this very first part, and then you'll jump into stage 3 at the beginning of the next video.